Lorne Brown here from Conscious Talks, and I'm with Dr. John McMichael from Beech Tree Labs. Now, John to me is an interesting character because of what he's doing in our world. I met him at a conference. I think, John, the discussion was on maybe brain injuries, which we'll digress to at some point. But because we're recording this in the area of COVID-19, I wanted to talk about that as well and just introduce some of your background. So it says here in your bio that you received your Bachelor of Science in Microbiology from the University of Maine and your PhD from Oregon State University in Immunology and Virology. And you did your postdoctorate work at the Wistar Institute in Philadelphia. You developed the world's first live virus fish vaccines for use in fish population and is an Alzheimer's disease pioneer. You've licensed over 30 plus patents and to support the Alzheimer's Initiative. And you now hold more than 200 patents worldwide. And your experience includes, this is why I think I'm so excited to talk to you, because you got some really interesting background education. Your experience includes professor of biology and health sciences, general manager of immunotherapeutics, where you license veterinary anti-inflammatory product, vice president of Molecular Prescription Incorporated. It's a biopharmaceutical company focused on products for managing inflammatory responses. Founder and president of Milkos Laboratory, a company now focused on botanical agents for nutraceutical use. And founder and president of the Institute for Therapeutic Discovery, a nonprofit organization focusing on the interplay between biochemistry and biophysics in the course of management of health. Now, I had to share this because I want people to know that you have kind of a really cool conventional background. Like you've done some really good education, and you founded Beech Tree Labs, which it says here strives to address unmet medical needs affecting the life of millions of people for more than 15 years. And you've developed formulations that target the body's signals to restore a healthy equilibrium. So John, thank you very much for making the time to be here. I want to get right into COVID-19. Now, looking at the news on your site, it looks like that you started doing trials on one of your products for COVID-19. Is that true?
Yes, that's right. We have a clinical trial going right now in Salt Lake City. This is interesting in that it's an outpatient trial. We're looking at symptomatic patients. We want to keep them out of the hospital. It's all done by telemed. That is, it's a virtual trial. We never have the patients come into the clinic, and we don't go visit them. We can do it all over a video on iPhones and things like that. We make contact with the patients every day, make sure they're taking their drops. We can measure their blood pressure, their blood, the oxygen saturation of the blood, their body temperature, things like that every day, telemetrically, virtually, so to speak. So it's kind of an exciting trial. We've never done one like this, but it is underway right now. It's a 40-patient, double-blind, placebo-controlled trial. Half the people will get placebo, half get the real stuff, which we call TML, and that's it.
The product that you're using for COVID seems to be a product that you've had for a while that you're using for the herpes virus and influenza, so it's a viral product. But I was reading that it's doing something with the cell signaling. So for the public and for myself, can you kind of explain how this is, some people call this an unorthodox or not the typical drug method. And so can you kind of do your best to explain how this possibly is working?
Yes, we have a pretty good idea how it works. It is a little bit unconventional in the way it started, but that's why we're jumping through the conventional hoops with the FDA to let people know that it's real and that it can work. But to get back to your point about how it works, it does 2 or 3 things. We've shown that it does have antiviral activity directly. That is to say, it inhibits virus replication. It does not do this for all viruses. You mentioned 3 different ones. You talked about the COVID, of course, the herpes, and the influenza. And it works for all 3 of those. We have lots of information about all 3, but there are other kinds of viruses it doesn't do anything for. Just as an example, we've had very good luck treating true influenza. That's the influenza that'll knock you out of work for a week or 10 days. We see very dramatic responses within a matter of a few hours in terms of decrease in symptoms. It doesn't do anything at all for the para-influenza viruses. Those are the 24-hour flus. It doesn't touch them. We don't know why. going back to the COVID-19, besides its activity with respect to inhibiting virus replication, we also know that it acts at the gene level of the patient, in the sense that it stimulates what are called anti-inflammatory genes, so they do their work while simultaneously inhibiting pro-inflammatory genes. As a consequence of that, we see a fairly quick resolution of symptoms, and we avoid what's called the cytokine storm that leads to the real serious problems with these kinds of virus infections, where the immune response goes out of control and causes way more troubles than it cures. So we avoid the cytokine storm, we get the genes going back into balance of the way they should be, and we also inhibit the virus replication.
And are you able to discuss a little bit about how all your formulations kind of work on the term you guys use is resonant molecular signaling, and how this signaling or communication impacts disease as well as health?
Yes, the whole idea that we're working under for our platform is that diseases, especially chronic diseases, and clearly COVID is not a chronic disease, but these diseases reflect either misinformation, miscommunication between cells, or lack of communication altogether. And the consequence of that is dysregulation. And so you get the body doing things or failing to do things that it should. All we try and do is get things back into balance. That's the whole name of the game. So for example, with the COVID, as I mentioned, we try and downplay the pro-inflammatory genes and upregulate the anti-inflammatory, get things back under control. And the medicine itself, in this case, it's called TML for the COVID, simply acts as a signal. And it's fair to ask, well, what's the nature of this signal? And the response is, we're not sure exactly. But of interest is that we're confident it's a signaling mechanism for several reasons. One is there's no relationship between mass and dose. So if we have a COVID infection of a horse or a rabbit, they would get the same dose as the human. And they would expect the same response in the same amount of time. And we've done this with TML, the anti-COVID medicine, with herpes infections. In cows, horses, sheep, humans, and rabbits all get the same dose. It's simply acting as a signal. And the signal, again, is focused simply on getting things back into balance, making the assumption that the machinery is still there to be kicked back into balance. If there's an elimination of the machinery, we're out of luck. But if the machinery is there, we think we can get under control very specifically without any, for example, without any broad spectrum immunosuppression, without any adverse effects.
COVID is a acute condition. However, as in it's only been around for a little while, so hence acute, but there are a term for long haulers where people who have recovered from COVID, as in they're not positive with COVID virus, are still experiencing periodic fatigue, neurological symptoms, shortness of breath, and other things. So looking to the future, based on your background in immunology and viruses and what you've been doing, what do you think of helping these people that are being called long haulers?
That's a great question, and it's certainly a new one, a new question, since the long haulers are just recently identified. My take on it is that our anti-COVID-19 medicine that we're evaluating now in the FDA trial would help those patients, but we might need a second or even a third molecule to go with it to take care of some of those long-term symptoms, because it might very well be a different disease. I don't mean initiated by a different organism, different virus, but the pathophysiology might be broadened to the extent that we would need another molecule or two to get different aspects under control. That would not be surprising.
Thomas Hatch said the drug is not meant to cure those who already are hospitalized or in critical condition. It's meant to catch the disease early so a person does not end up in the hospital. That's what you're doing with your research. But again, if this is a signaling thing, this is all speculation because you haven't done a study on this, but what about the people that have a severe reaction and are hospitalized?
Again, I think we can help them, but it might take more than just the TML by itself. Okay. Because we might have to address the damage that the virus has already done, not just address the virus.
On the site under news, and you brought it up, that this one, the TML, was actually originally being used for the herpes virus. And but it sounded like it had been sitting on the shelves. So did this ever make it to as a remedy that people can get that are using for cold sores for the herpes virus?
No, no, we did 2 successful trials using the TML for what we call recurrent oral herpes infections. One trial was focused on patients that had a history of oral herpes outbreaks after dental procedures. And the second trial was focused on patients that had outbreaks after exposed to UV light, like being out in the sun in the wintertime or on the beach or whatever. The trials were successful. We, as measured by patient response, lack of adverse effects, and p-values, statistically significant p-values. We ran out of money and to do what's called a phase 3 trial. Those were 2 phase 2 trials. We didn't do a phase 3, and then the patent got old, and so it became unattractive to potential licensees. So it's out there. The physicians and the veterinarians in our group used and continue to use the TML for a variety of herpes infections in different species, not just recurrent oral herpes, but it has not made it clear through the FDA sequence of events to be on the market.
You have the TML now being studied for COVID, and then any other products in the pipeline for conditions?
Yes, there are several. As you mentioned in your introductory remark, the next in line is one for treating chronic traumatic brain injury. This is really important to us. We've had a lot of experience treating patients who have had brain injuries from falls or athletes that get their head beat up like the NFL players, car accidents, falls in the home, things of that, and abuse patients and so forth. And we find that patients respond to this therapy in terms of improved memory, cognition, control of their emotions, ability to go out and work again, even if the injury occurred decades before we start the therapy. So this is one that we're really anxious to get into FDA trials, but everything has been set aside because of doggone COVID. Seems to garner all the attention right now.
And what's the mechanism behind this one? Because you talk about cell signaling in COVID, you talk about the inflammatory response. If somebody's had a brain injury, is it the same mechanism or is it a different mechanism?
It's doing 2 or 3 different things. Again, it's a different formulation. It's not TML. It's altogether a different formulation. And it decreases inflammation in the area of the injury, and that's a chronic problem in a brain injury. So we decrease the infiltration of the cells that are associated with the injury. We are able to get rid of gliosis, which is kind of the brain's form of scar tissue. And it appears, we haven't proven it yet, that it increases blood flow in the area, which would again improve oxygenation and also get rid of the junk, the debris that's there from the damage. So it does at least those 3 things. 2, we're confident of. The third, the blood flow. We have a mechanism in collaboration with another group to measure that, but we have not done it yet.
NGF, the one on anxiety and depression.
Yes.
Where is that in the pipeline? And I think of that one because it just seems to be epidemic anyhow in our society. And I think quarantine and COVID-19 has really brought that to the surface for many.
Well, it sure has brought those problems to the surface. It's back in the pipeline of ways because of a matter of resources for us. But of interest, you're interested in mechanisms of action and so forth, how it works. We did a really nice series of studies with collaborators at the University of North Carolina at Chapel Hill, who are experts in mechanisms of actions of anti-anxiety and antidepressants. And what we found and published was that the NGF works in a fashion completely different than anything on the market, but we don't know how it works. We know how it doesn't work. We don't know how it works yet. But it is really interesting that it does, and it works in animals like neurotic dogs that get afraid when the thunderstorms come, or they have anxiety when the owner goes away and they end up chewing up the furniture and that sort of thing. It works in animals as well as it does in humans, but we're not sure of the mechanism of action.
And so if you're not sure of the mechanism, how did you figure out this could possibly work for anxiety, depression? How did you guys even think of using or creating this product?
That's a trick question. I've got to think about just how that happened. And as usual, it was an accident.
You figured.
We thought that this might work for dementia, or at least as a component of dementia. And we found out that in some younger people that had no history of dementia, but other than in their families, and they wanted something that might prevent Alzheimer's for them in the future, whatever. Had a couple or three patients like that, and we tried it just for the heck of it. And if they were anxious, they got better. And so it was not planned. It was good clinical observations by the physicians in our group. And then expanding on those observations, we were able to do the work at Chapel Hill that I mentioned, and also to look at it even helps with addiction. In fact, one of the early patients that we used this low level of NGF for was a nurse that was having problems. She was stealing medicine from the hospital pharmacy. She was addicted. She knew she had trouble. She came and asked one of our physicians, is there anything that he could do? And he said, well, let's just try this. By golly, it worked. And so again, it led us from one thing to another.
Excellent. It seems like your products and your delivery is microdosing, like so less is more. So am I correct in understanding that?
Within limits. It's not microdosing like homeopathy, for example. It's not nearly as dilute as that. It would be microdosed compared to a lot of allopathic medicine. But if you think it, there's what's called hormesis. I don't know if you've ever heard that term. Hormesis. And hormesis refers to the fact that let's just take a toxin, let's take radiation, okay? Radiation can be toxic, we know that. You get too much of it, you're gonna be in trouble. If you look at a typical radiation curve or a typical curve for toxin, it goes this way. The more toxin, the more lethality, the more serious the complications. If you go way down to the bottom of that curve, there's a point where the toxin can actually be beneficial. That's the hormetic window, where a molecule that at one concentration is going to cause a problem, another concentration will not. Give you an example again with radiation. We think that people that work in the radiation field, being exposed every day or 5 days a week, would probably have an increased incidence of different kinds of cancer because of that radiation exposure. But that in fact is not the case. In fact, in many respects, they have less cancer. They're seeing a hormetic effect. More recently, radiologists are treating some sorts of diseases with less radiation than they used to and getting as good or better effects. So there's this U-shaped curve. And if you get at the bottom of that window, you can do some pretty amazing things pretty quickly. For example, you had mentioned again the TML being used for influenza. And TML at high concentrations can actually be a toxic, can be an antimicrobial substance. At the low concentrations, it loses its toxic ability, but in a person that has the true flu, the one that's going to knock you out, we'll see a diminution of symptoms within 45 to 60 minutes. That's pretty quick. You would never get that. You wouldn't get any response at all in a concentration 10-fold higher and 10-fold lower, you also would not get any response. You've got to get in that window. And that's what we try and find is that hormetic window.
With your EML, the one that has been used for viruses, the herpes and influenza, and now being tested for COVID, it looked like when I was reading some of your publications that I looked like the success rate maybe was close to 50% with the patients you had. Did I read that correctly?
No, it's much higher than that. I think it's fair to say that looking at it anecdotally, not controlled studies, that it's well over 75%. With the controlled studies, like the herpes studies, it again was well over 50%. And that, well, we had a p-value of 0.0297. So that's pretty nice distinguishing between treated and placebo. So you're well over 50% in terms of the number of treated patients that responded positively.
And it's not a fair question, but it's a curious question: is Why does it not work for everybody? If you're doing it on a genetic level, a cell signaling level, low dose, why is it that some people won't respond?
So that is a fair question. It might be that they need a different dose. It might be the frequency of dosing is different. I'll give you an example. We have a formulation that we use for arthritis. And I can remember a lady that was taking our prescribed protocol at the time, was take a drop 4 or 5 times during the day. She had taken the drops as recommended for a good 6 weeks and saw nothing at all, and was really discouraged, and rightfully so. And I said to her on the phone, and her physician was on the phone with us, a 3-way call. I said, listen, before you give up, let's just try something. We're confident this stuff is safe. We've jumped through all those hoops. Why don't you just try a drop every hour for a day, just for fun, and see what happens? She did. She called back the next day, and that was it. Now, that her arthritis wasn't all gone, but it was much better. She did that for 3 or 4 days, really frequently like that, and then was able to go back to 1 drop 4 times a day and maintain that improved status. So maybe it just takes more to kick some people into gear. I don't know. That's just anecdotal, but we've seen that on more than one occasion.
But you said earlier though the dosing didn't matter based on age and weight. So dosing does matter, or does it not matter?
Well, the concentration doesn't matter. The amount of material, of active material in the drop doesn't matter. But yes, in a sense that's contradictory. The frequency seems to matter more than the concentration. For example, taking 1 drop 4 times a day does not equal 4 drops once.
When should we expect phase 3 with this with the TML? When is that expected?
Well, I thought it'd be all done by the end of June, and here we are in the first part of September. The gating factor is rate of enrollment of patients, and we've been very slow in getting patients in. As a consequence of that, we're going to open up, I think, 1 or 2 more sites, 2 more locations. But to answer your question, I don't know, but I would hope within the next 4 weeks we have all the patients in there.
And how many patients will be in phase 3? Do you need?
For phase 3, we're probably, for phase 2 that we're doing now, we're talking 40 patients. I think I mentioned that.
Okay.
For phase 3, we're probably talking 250 to 300.
Okay. So you're right now recruiting for phase 2 for the 40 patients.
That's right.
I'm surprised it's such a challenge to get patients in, considering there is no shortage of COVID positive people.
Well, you're no more surprised than we are. We thought it would be a walk in the park. But I think the biggest problem is getting the information to the patients that there's a clinical trial available. And that's not been easy, because remember, these are outpatients. And our criteria now are they have to be 55 years older, and they have to be symptomatic. They have to have symptoms. And they have to have a fresh, that's within 48-hour positive diagnosis with the COVID test. And I don't know if that's limited them, if we've limited our population unnecessarily, we just don't know. Although I can tell you within the last, I think, 10 days, we've started some social media advertising and seen a tremendous increase in at least the number of people coming to the website. And I would hope that will translate to people that find that they are eligible and are interested in enrolling. And I think, we are not well known as a company. Who ever heard of Beech Tree Labs? So it's not as if it's a Pfizer or a Merck. But they can go to clinicaltrials.gov and see that we are a legitimate FDA authorized trial. And it's a matter of people having confidence that we have something to offer, that we're legitimate, and then getting them in there. And I think with this social media approach, it appears that we're gaining some ground pretty quickly on that.
And I want to share the website is beechtreelabs.com, BEECH treelabs.com. And my understanding is there's opportunities to donate and there's opportunities to invest. So do contact them at beechtreelabs.com. You can see their peer-reviewed publications on their site, the news, and the product lines that they have coming. The anxiety one, again, is that one in the pipeline? Is that being researched yet? You mentioned that, but I can't remember what you said
Yes,
where that was.
We're continuing our research, but again, because of limited resources, it's in the pipeline, but it's not front burner, it's back burner. the next front burner one is the traumatic brain injury one.
Okay. Can you explain how that,
Sure.
what's in your product versus in vaccines and safety issues, et cetera?
It's the same material.
Okay.
The TML stands for thimerosal. That's what it stands for. So it's the very same product that was in vaccines, or still is in vaccines. It's about 125 times lower concentration than in a vaccine. And in fact, if you look at what the World Health Organization allows for daily exposure all your life to methylmercury, which is more toxic than ethylmercury. TML is ethylmercury. If you look at the daily allowed limit from World Health Organization, the daily allowed from the EPA and from the FDA, you will find that if you take all of the mercury exposure collectively from our protocol for COVID, it adds up collectively to about 1/10 the daily allowed dose from those other organizations, and it's methyl versus ethyl, with the methyl being more toxic. So we're so low in toxicity that the FDA said, don't do any tox studies. We know this molecule, and at this concentration, we don't know if it'll do anything positive, but we're confident it's not toxic.
Is it being used as a preservative that is usually used in a vaccine, or is it actually the active ingredient?
It is the therapeutic molecule.
Okay. And so the follow-up question around that is, there's so many people that have concerns with vaccines and say it's the thimerosal that's causing all these problems. Based on your background and your research, what's your thinking then and you've created vaccines between TML and the thimerosal in the vaccine? Is it the same concentration dosage, or is there a different?
No, we're over 100 times lower concentration.
Okay.
I think it's a legitimate concern. If a person is concerned about vaccines, what is the role of the thimerosal in the vaccine? Especially some years ago, the question was, is thimerosal causing autism? That was the big problem. There's been lots of science to show it doesn't. The autism level has not receded as a consequence of removing the thimerosal from vaccines. So that gives you a little bit of a hint. And again, it's a matter of concentration. At one concentration, thimerosal is a potent antimicrobial. That's why it's in the vaccines, to keep them clean of microbial contamination. At the concentrations we're using, it has no antimicrobial action. It's worthless as a antimicrobial, but it's dynamite as an antiviral against some viruses. So again, it's different concentrations of the same molecule getting different biological effects. And as indicated with the 2 controlled studies we did using the very same protocol we're using for the COVID, the 2 controlled studies with herpes, the placebo patients had more adverse effects than did the patients getting thimerosal. And neither arm of the study had serious adverse effects.
Thank you very much. I really appreciate taking the time. Again, this is Dr. John McMichael from Beechtree Labs. Their website is Beechtree Labs. That's 2 E's, B-E-E, beechtreelabs.com. Do you have any final messages, anything you want to communicate, any questions that you want to put out to the public?
Only that if you're in the Salt Lake City area and want to become involved and have COVID-19, you're an outpatient and interested in clinical trial, there's a website for that called sickwithcorona.com. And I'm sorry, with coronavirus, sickwithcoronavirus.com. The only other thing is I would like to thank you very much for the opportunity. It's a pleasure.
My pleasure. Thank you for making the time. Oh, last question that will fit in is, I guess, 2-part question. Can you take it preventatively, prophylactically, 1? And 2 is, if you know you've been around somebody, is it something that you would take just in case?
Yes, that's a great question. And I can tell you the physicians in our group that are around COVID patients take it prophylactically. And they have not come down with COVID. Now, we've done no clinical trial. We're focusing on this one now without patients, like we mentioned before. But it seems to me that it would be some sense to do a formal trial to answer that very question. Again, anecdotally, it looks like it might work as a prophylactic, but we haven't proven it yet.
And if you were to do a study based on what the doctors are doing, what kind of, is the dosage the same as if somebody was active with COVID, or do you change it?
We'd change it to taking 1 drop twice a day.
Okay. Dr. John McMichael, thank you very much. I look forward to having more conversations with you. Again, check out their website, contact them if you are interested in getting involved in trials. If you're interested in investing and donating, do check out their website. And again, thank you for your time today.
Thank you. It's been a pleasure. Take care.